All right. All right. David Ress was initially trained as an electrical engineer at UC Davis. In 1980, he got his BSEE. He then made a transition to study plasma physics, and during his graduate work at Stanford University, he focused on magnetic fusion experiments at Lawrence Livermore National Labs in Livermore, California. He got a PhD in his EE in 1988, and he continued working at the Lawrence Livermore Labs, but he was a bit unhappy about the direction. So he decided to change to study neuroscience, because he knew that he could apply his wonderful education and his great brain power to doing something very positive for the world. So he studied neuroscience, using the methods of electron tomography and functional magnetic resonance imaging, with the latter eventually becoming the main focus of his work. During this time, his research focused on the use of high resolution and sensitivity methods to investigate neuroscience issues in human vision. And he became an associate professor at Brown University in 2005, where he played a key role in developing an MRI facility for their program. He then moved to the University of Texas in 2007 and developed their imaging research center and performing research in high resolution MRI and modeling of cerebral vascular physiology and physics. He moved to Baylor College of Medicine, which is in Houston, and that's where he is now. He moved there in 2013, and he's continued with similar subjects, and he's working as technical director of the Center for Magnetic Resonance Imaging. So now I'm going to turn it over to David, if you'll share your screen.
Hello, everyone. Thanks for coming and listening to a bit of a talk about measurements and modeling of the hemodynamic response in the human brain, a topic that's a little different than most applications of functional MRI, which are basically aimed to study brain activity to try to interpret aspects of cognitive science. Here, we're actually more interested in why the brain is doing this interesting process that gives rise to functional MRI, and try to interpret it a little differently so that we can eventually learn about aspects of pathophysiology. So I'll start with an introduction to functional MRI. I'll cover nuclear magnetic resonance and the signals that we get from it, then talk about about this phenomena called neurovascular coupling, and that's the basis for functional MRI. And one of the key signatures of functional MRI is this hemodynamic response function. It's really a blood flow response that's measured in functional MRI, not the usual notion of electrical activity. That's been the gold standard in neuroscience. Then I'll give you a bit of a taste of how one can model the hemodynamic response. And I'll conclude by talking about a few sets of measurements of the hemodynamic response, specifically whole brain characteristics, because we have techniques that can evoke the hemodynamic response across the whole of the brain in one scanning session. And then I'll talk about the temporal stability of that response and give you some applications where we're studying age-related pathology. So let's talk about MRI and then functional MRI. So starting at the beginning, if you place a tissue sample, and specifically the tissue of interest is water-based, and MRI works best with aqueous samples because those are the most prolific and strongest of the nuclear magnetic resonance signal sources. So when you put a big sample full of protons, the proton spins become oriented. Some are parallel to the magnetic field, and some are anti-parallel, and the number that are parallel is slightly greater than the number that are anti-parallel. And the result is this net magnetization. This net magnetization is the fundamental source of signal in magnetic resonance imaging. So there's an energy difference between the parallel and the anti-parallel states. The parallel states are in a lower unexcited state, and we call these hydrogen molecules spins because we're manipulating their quantal spin state. But we can flip their state from parallel to anti-parallel by applying an RF magnetic field perpendicular to Z. So we can drive, using magnetic induction, we can drive excitation. So here's a bit of a graphic about that. So in equilibrium, we have our orange parallel states, a somewhat greater number than the blue anti-parallel. We then apply an RF excitation, and that flips some of the spins into the excited anti-parallel state. And we use a pulse of RF applied by simple magnetic induction. And the frequencies involved are at 3 Tesla is about 128 megahertz. 7 Tesla, it's closer to 300 megahertz. And after you turn off the excitation, you get emission of the RF at the same frequency. And that's basically an echo, and it's the most fundamental echo that you can get in magnetic resonance imaging. It's called the free induction decay. Now, when you excite the spins, if you use a so-called 90 degrees pulse, so that all of the longitudinal magnetization that's available is reduced to zero, so-called saturation, then the signal recovers, the longitudinal magnetization recovers quite slowly. The time constants are on the orders of seconds. So MRI is kind of a slow process, which is why you have to lay in your MRI machine for so long when people are taking images of your brain or other tissue. But the excited signal, which appears in the transverse plane, decays much more rapidly. The time constants are on the orders of about 100 milliseconds, 10 to 100 milliseconds. And that time constant is either called T2 or T2 star. And it's the most important one for functional magnetic resonance imaging because we use so-called T2 star weighted imaging to measure brain function, because that's the decay constant T2 star in specific, that's affected by blood oxygen level. We'll talk about that in much more detail. So there are fundamental contrast mechanisms that you can get out of MRI. The most fundamental contrast mechanism is called proton density contrast, where you avoid all relaxation mechanisms, and then you just measure as best you can the longitudinal magnetization throughout the tissue. So this is the most fundamental MRI signal and image measurement. or you can weight T1 by emphasizing the longitudinal relaxation effects and minimizing transverse relaxation effects. Finally, there's T2 or T2 star weighted, where you emphasize transverse relaxation effects and minimize longitudinal relaxation effects. And T2 star weighted imaging is what we're going to be most focused upon tonight. So MRI image acquisition is a neat trick. Usually in functional MRI, we use 2D acquisitions where you start by doing a slice selection that creates a single plane of excited magnetization. And we want to resolve the MR image on this slice. And the slice image detail is acquired in the Fourier transform domain, the so-called K-space. And what you essentially are doing in acquisition is that you acquire K-space by applying phase modulation using the gradient coils. So all that thumping noise that you're hearing when you're laying in the MR scanner is this gradient, or these gradient magnets, which are applied, which are essentially electromagnets that are time-varying electromagnets, apply a gradient signal that essentially creates a phase modulation that sweeps you through K-space. And in most imaging protocols, most anatomical imaging protocols, the image is acquired very slowly because we acquire it slice by slice. And we have to acquire, pardon me, not slice by slice, but we do acquire slice by slice, but we also acquire line by line. And so we use the electromagnetics, the gradient coils, to sweep ourselves through K-space and just acquire each thump of the magnets is just a single line in K-space. And then that gives you one image. And you can interleave the images to some extent because you can acquire it. You can interleave your slices, but it still takes you a few minutes to get an image volume. In contrast, in functional MRI, we use a different trajectory that sweeps usually back and forth or sometimes circularly through K-space to acquire all of the K-space information at once on that excited slice. And that way we can get the speed up to the point where it only takes, say, 1 to 3 seconds. There are some acceleration schemes that can push it down to as small as 1/10 of a second. You acquire all of the K-space information, all of the necessary K-space information. And then once you have the K-space information and you've done appropriate regularizations and the like, all you need to do is transform it to get your image. Now, where we get into functional MRI is that it turns out that blood flow affects the MR signals. So blood is an onboard, an endogenous contrast agent because oxygenated hemoglobin is almost non-magnetic, but deoxygenated hemoglobin is a little paramagnetic. So these differences in susceptibility cause changes in the image brightness because they change the transverse relaxation rate. So here in this picture, we're going to excite our slice of spins. So everybody, all of the spins are spinning synchronously, coherently in phase. And then there's going to be decay, this T2 star decay, which is induced to a large extent within and adjacent to the blood by the hemoglobin magnetic field effects. And then after waiting a while, our so-called echo time, we acquire the image. And the oxygen dependence is such that when the blood is well oxygenated in a voxel, there's a little more signal than when the blood is deoxygenated. So generally, we can make use of the fact that blood oxygen level changes during brain activation. But the basic idea here is that deoxygenated blood is dimmer, gives less signal than oxygenated blood. So here's another example. Here's a more quantitative picture of how oxygen concentration affects T2 star. So for oxygenated blood, the T2 star is very long. It's a quarter of a second. Deoxygenated blood, fully, almost very, very lightly saturated hemoglobin, has a T2 star that's down around 17 milliseconds. And so this big difference in T2 stars can result in a big shift in the image brightness. It's also good to know that the longitudinal relaxation rate is independent of the oxygen concentration. So you only need to worry about the change in the transverse relaxation time. So here's an example of how this plays out in tissue. So we know that deoxygenated blood has a shorter T2 star, a shorter transverse relaxation time. So the signal decays faster and it will appear darker in these T2 star weighted images, these gradient echo images. So here's an example of a rat brain, an image of a rat brain, which is this thing in here. And here's a rat brain that's a rat who is inspiring normal air. And here's a rat that's in hyperoxia. And what you can see is because this state of hyperoxia has caused the blood in the veins to become rather highly oxygen saturated. The veins stop being dark. They essentially turn white, almost white. And so this is sort of on a global scale what is happening, the contrast mechanism that creates functional MRI. So here's the functional MRI cartoon. Neural activity causes oxygenated blood to flow into the activated region. This is an interesting phenomenon that we'll talk about a little bit later. It's sometimes called functional hyperemia, but it's a very clear process in the brain. Active brain tissue gets more blood, and it's an interesting and complex sequence. We'll discuss in more detail. But the oxygenated blood flow, this increased flow, displaces deoxygenated hemoglobin. And the displacement of deoxygenated hemoglobin causes T2 star to go up. The fact that T2 star is longer means that there's more signal. There's a brighter image in that region of the more signal in that region in those voxels. So the active brain lights up. So this is a spatiotemporal phenomenon. So in arteries, the blood is always highly saturated. So it doesn't change whether or not the brain is less active or more active. But in the capillaries that feed the active area, there's greater blood flow, and so the oxygenated blood washes in to the capillary parenchyma, and that pushes out the deoxygenated hemoglobin and makes a voxel that is emplaced upon, that contains these capillaries, have more signal. It also makes the signal brighter in downstream veins. So how do we use this interesting bit of mixture of physics and physiology to measure brain function? So we have to think about this cascade of processes. The ones that we can manipulate in experiments are sensory, motor, and cognitive processes. We ask a subject to look at things, to do tasks, to move in various ways. They can't move very much because they have to hold their head still, but we can ask them to do things. Sometimes they can tap their hands or their fingers. And these demands of stimulus and task result in integration and signaling in ensembles of neurons. Those neurons now, that signaling requires ATP consumption. And the usual notion about functional hyperemia is that the consumption of that energy requires blood flow, but it's actually a bit more complicated than that. So the blood flow brings in oxygen directly and glucose as well, and displaces deoxyhemoglobin, and that is what creates our functional contrast. And the brain is really superbly designed to deal with vascular responses. So the brain is one of the most blood demanding organs, and that's the usual notion. And it is. But the brain's consumption of oxygen and sugar is kind of like a moderate usage of your quadriceps. When you really push muscles, they're even more demanding than a brain. But the brain's not really doing anything except doing processing, except signaling for the most part. So it's kind of like a signaling muscle. So how is this done? How is this functional hyperemia managed? And this picture at the upper right shows an endovascular casting. So the endovascular casting is from a rat, and you make endovascular castings by just before you sacrifice an animal, just as you sacrifice an animal, you wash out all of their blood and replace it with epoxy. And then you let the epoxy set, and you dissolve away you cut a slice, dissolve away all of the tissue, leaving the epoxy replica of the vasculature. And then you coat that with heavy metals so you can take a scanning electron micrograph. And this is what they look like. So here you can see a peel arteriole, and you can see coming off of the peel arteriole are a couple of penetrating arterioles that are diving down into the capillary parenchyma below. Next to the peel artery is a vein. You can see a vein here, and there are also venules that come back up to return the circulation. But the vasculature is very interesting here because this in-out is strictly by way of the top through this peel mesh. And there are control structures to regulate this flow of blood. What you can see in the endovascular casting is that some of this striation here, these are smooth muscle fibers. This is not a perfect replica of the way they look in vivo, of course, but you can see them here. And these smooth muscle fibers can adjust the diameter of the arterioles. And the smooth muscle fibers are also present in the penetrating arterioles until they penetrate a few hundred microns into cortex, and then the smooth muscle fibers go away and are replaced by other regulatory cells called astrocytes. But there are lots of experiments that show that blood flow control is present all the way down to the capillaries, and maybe there may even be flow controls in the capillaries. These pericytes is another kind of cell in the brain that is associated with flow control. The pericytes have end feet that wrap around the blood vessels. Sometimes the end feet are filled with the epoxy, and you can see these structures wrapped around an arteriole. And sometimes you can see the indentations left behind by the pericytes. So this combination of smooth muscles, individual myocytes, pericytes, and maybe more intricate flow control mechanisms such as the glycocalyx, this sticky material that's exuded on the insides of blood vessels, control the flow very, very accurately. And the flow control is in this functional hyperemia that occurs when activity happens, is specific to about the scale of 1 millimeter. So if a very small region of your brain becomes active, then a little 1 millimeter region of blood flow will become enhanced. So that results in neurovascular coupling. And the dominant hypothesis about how neurovascular coupling is initiated is called proximal integration. So the notion being that down in the capillary parenchyma, where the neurons are adjacent to the capillaries, there's some amount of activity. It turns out that the activity that's most sensitive that drives the blood flow response predominantly is excitatory synaptic signaling. When that occurs, there is a propagating vasodilation. The small arterioles, maybe even the capillaries, dilate, and that dilation spreads up the tree, the vascular tree, all the way up to the pial arterioles. And if it's a very strong, broad region of activation, the dilation can spread all the way up to the big arteries that feed the brain. So you get you'll end up with a big pressure fluctuation that will then result in downstream flow changes. And there's much that we don't know about neurovascular coupling, but this is the best picture that we have so far. So what does this look like to functional MRI? This hemodynamic response, the so-called blood oxygen level dependent hemodynamic response to a short stimulus, has this form. And we call this response is called the hemodynamic response function, because if we're going to treat the system, the brain MRI response, as a linear system, which we tend to do in order to interpret brain responses, then this is the impulse response. This is the point spread function for functional MRI. So here are a couple of measurements I took long ago in an early visual cortex. There's a 2-second visual stimulus that occurs between here and here in time. And what happens, so the electrical activity that you get from a 2-second visual stimulus is pretty much confined to the period of the visual stimulus. Once you turn the visual stimulus off, if you look at signals in primary visual cortex, or most of the immediately adjacent extrastriate areas, that electrical activity will simply stop when you turn off the stimulus. And the bold response, this hemodynamic response function, during that period of time that the stimulus is on, nothing happens, except maybe a tiny little tip. Then the signal, a few seconds later, the signal starts roaring up until it increases a substantial amount. And this is, it's not a huge amount because there isn't that much blood in the voxel. The fraction of blood in a gray matter voxel is only about 5 or 6%. But that 5 or 6% does change its oxygen saturation substantially. And so overall, we see something on the order of about a 1% change. Okay, and that 1% change has a time delay of about 6 seconds, the so-called to the hyperoxic peak. And then after this peak in the signal, we have an undershoot. The signal will generally go down below baseline. And then there does seem to often be some amount of ringing before it goes back to baseline. So this is the character of functional MRI. We take electrical activity that's very prompt with the stimulus and has time scales of milliseconds. If you look at spiking activity, the time scale of a spike is on the order of a millisecond. The time scale of the signal that a spike creates on a downstream synapse, those are called postsynaptic potentials, they have time scales of 10 milliseconds. And so we have 1 in 10 millisecond sort of electrical time scales, but now we're going to try and characterize those using this sluggish response that doesn't reach a peak until about 6 seconds after the response, after the stimulus begins. So this slow response, delayed and slow, is what we have to work with. You can model the hemodynamic response function if you, and the model is actually kind of appealing. So we developed this thing called the arterial impulse model. This was the work I did in collaboration with Jung-Hwan Kim. And this model postulates that there are 2 inputs to the system: the blood flow response, which is described using a linear network model, and an oxygen metabolism response, the demands of the cells, which we're going to describe just using a simple gamma function. Then we link these to the bold response using a differential equation description of vascular oxygen transport. And this is going to be a 1D compartmental model. And then we can use a standard linear relationship between the bold signal and the intravascular oxygen concentration. We know it's actually exponential, not linear, but the changes are very small, so we can approximate this as being quite linear. And we're going to divide this into, divide this into 3 regions. An arteriole, where there's no oxygen transport, but this is where the flow response is generated. And then a capillary, where the oxygen transport occurs, and a vein, where there's no oxygen transport and no volume change. So the blood flow response we describe using a 4-element linear network. The compliance in this. So this 4-element network has here's an electrical analogy to a fluid system. And it contains an inertance, resistive elements. One resistor is briefly shorted out to simulate vasodilation. And then there's also a compliant effect. The compliance consists is created by the elasticity of those smooth muscle fibers that are in the capillary, in the arterioles, the PL arterioles. And then the parenchyma itself is simply resistive because the dimensions of these blood vessels are tiny. They're on the orders of 20 microns or less. So if you take this linear model, then you get the solutions are simple. Either they're underdamped, a ringing response, an underdamped sinusoid, or they're overdamped. And although typically we find that the model is best described, the signals are best described by underdamped responses. So oxygen transport gives us the bold response. So the oxygen transport assumes that there are 3 compartments. There's an erythrocyte compartment where the red blood cells are. Plasma, where the red blood cells leak their oxygen from the hemoglobin into the blood plasma by way of a dissociation process. That's how we lock up the hemoglobin dissociation. And then there's diffusion across the blood vessel, the capillary wall, into the tissue space. And then there's oxygen uptake by metabolism in the tissue compartment. And so if you write down this set of coupled partial differential equations for the 3 compartments, then you can predict the oxygen transport. And then once we have these extravascular erythrocyte and plasma oxygen concentrations, we can now relate those now to the bold signal in terms of there are 2 pieces. There's an intravascular component, that's just local dephasing within the blood vessels, and then there's an extravascular component that corresponds to dephasing that's from the blood vessels, essentially causing dephasing in surrounding tissue. And this simple linear relationship gives us an approximation to the bold signal. And we can use a gradient descent modeling procedure so that the flow model has a set of 3 parameters to describe it. The oxygen metabolism piece has a set of parameters that describe it, another 3 parameters. These go into the transport model, and we then get some sort of predicted bold response, and then we use gradient descent optimization. It would be nice to use a gridded search, exhaustive search to do the optimization, but at the moment the process is a bit too slow. We could make it faster, but we have the issue is that we have many voxels in every measurement, and so doing an entire brain by exhaustive search would be too slow. So here's examples of the quality of some of the fits. Measurement is in blue, the bold model is the aim model is in red, and here are 2 example subjects. and here's an average across many measurements. And here's what comes out of the model: predictions of the blood flow and the oxygen metabolism. And these results are very reasonable. And in fact, we actually measured the blood flow directly using a technique called arterial spin labeling, and the blood flow responses matched, including this critical aspect of an undershoot. So the blood flow response does indeed appear to match this assumption of an underdamped sinusoid. Okay, so now I'm going to talk about some particular HRF measurements. Specifically, we'll talk about whole brain characteristics of the HRF, and then we'll talk about how stable those HRF measurements are, and then some new work where we're trying to look at using these whole brain HRF measurements to look at age-related pathology. So we have developed a technique called the SAST, the Speeded Audiovisual Sequence Following Task, to essentially light up the whole brain with these hemodynamic response functions. And the SAST is a very simple task. You get, during a 2-second period of time, you're laying in the scanner, looking at the stimulus through a mirror behind the scanner bore on an LCD that's MRI compatible. And you'll see these 3 regions of colored dots, half brightly colored, half dark, for maximized contrast. And these 3 regions will go off in the last 2/3 of a second each, and they'll go off in a random sequence. So here the example is first yellow on the left, then red on the right, and then green in the middle. And what your task is to push a button on a little response cylinder that's in your right hand as the regions of colored dots, which we colloquially refer to as fireworks, as the fireworks occur. So it's not a difficult task, but it requires a lot of brain function to do. At the same time these visual stimuli are going off, there's also a bandpass filtered noise tone, so that you get it's highest pitch for the green, lowest pitch for the red, and a medium pitch for the yellow. And so you can pay attention to the color, or you can pay attention to the sound, or you can just pay attention to the position. It doesn't really matter how you do the task. You get this multisensory stimulation, and you have to do this button pressing task. And then after 2 seconds of this task, there's a 28-second period where all that happens is the little fixation dot changes color about every 6/10 of a second. And your task is to push a button, one of the 3 buttons, every time this turns yellow. Okay, and it doesn't turn yellow very frequently, so it's a very slow paced, and it's a very easy task. So we have a lot of action here, and then very little action here, and that creates our functional contrast. And with this task, we scanned 20 subjects, half male, age range of 20 to 64 years. And we used fairly high spatiotemporal resolution functional MRI. We used this acceleration scheme called simultaneous multi-slice acceleration, which basically means 3-to-1 means that you excite 3 slices in the volume at the same time, and so you acquire a superposition of 3 slices, and then you use a separation technique that's based on so-called slice grappa. It's based on a mixture of linear algebra plus some tricks in the temporal frequency domain to sort out those three slices. We also use 2-to-1 k-space interpolation to keep the echo time, down at 30 milliseconds, which will give us good functional contrast. And these were fairly high resolution measurements, 2 millimeter voxels. That's high for functional MRI. And the temporal resolution was, the sampling was 1¼ seconds, and 60 slices to cover pretty much the entire brain, at least all of cortex. We missed a little bit of cerebellum, but we're not that interested in cerebellum. So we get a lot of HRFs. We get HRFs over a large fraction of the brain, and we're going to parameterize our HRFs. So most of the HRFs look like either this, positive HRF, and we're going to extract parameters such as peak amplitude, full width half max, time to peak, and undershoot from this waveform. And some of the, interestingly enough, a fraction, roughly 15 to 20% of the activated tissue, of activated portion of cortex, shows up with negative HRFs, where the brain appears to deactivate. And we get a similar set of parameters from the negative HRF. Now, some voxels, some HRFs can show a bunch of high frequency response. And so we want to quantify these high frequency phenomena we use the parameter of high frequency power fraction, so that in the transform domain, the temporal frequency domain, we calculate the amount of power greater, our Nyquist frequency is 0.4 hertz. So we calculate the amount of power in the upper half of our measurement band. And that fraction of the power is going to be another parameter that we extract. Now, this simple task, the SAST, produces responses almost over the entirety of cortex. These hemodynamic responses look like these examples. Here are 5 examples, and they all look roughly like the example I showed you back when I was talking about functional MRI. There's this hyperoxic peak and then an undershoot afterwards. And the negative HRFs look a little different. They usually have a little bit later time to peak, and there's much less frequently an overshoot than there is an undershoot for the positive HRFs. And if we adopt a fairly high requirement for CNR, peak CNR has to be greater than 3, because we want to be able to have enough CNR to calculate these temporal parameters. We find that on average, we get 87% of cortex lights up. This is an average taken from 55 total sessions, where we've run this on a total of 34 subjects. Some of them were scanned more than once. And you'll see that the SAST evokes a fairly stereotypical amplitude pattern. There's strong signals in visual cortex here and here, strong signals in auditory regions. There's also strong signals in certain portions of somatosensory cortex, attention areas. And the this essentially this stereotypy of the amplitude response is which has a correlation across subjects, a mean correlation of 0.6. This shows the correlation between across the 20 subjects. With a mean value of 0.6, it's kind of like a fingerprint of what a healthy brain, how a healthy brain responds to a particular task. The timing doesn't really vary very much across subjects. So this is time to peak, full width half max, and time to undershoot. And the variations across subjects are only about plus or minus 10% on this 6.1 second number. Full width half max, likewise, maybe about 12% variation. And time to undershoot is the most stable of all. It's only about a 9% variation across subjects. So once again, the stability of the dynamics also suggests that this is the way a healthy brain should operate. So if it's different than this, then maybe something's wrong. There are some stable patterns of timing variations, which is interesting for a number of reasons. Here are timing variations, plus or minus, on a scale of plus or minus 1.5 seconds for time to peak on 2 subjects. So that you can see regions where time to peak is significantly later, and there are regions where time to peak is significantly early, here in this region of lateral frontal cortex, and here's across medial regions from parietal through frontal cortex. So again, this is kind of an interesting signature. And what's also interesting is that visual, auditory, and somatic motor regions exhibit faster and shorter full width half max, whereas early visual, dorsal visual, and attention areas exhibit slower and longer full width half max. We think this has to do with the amount of oxygen metabolism that's occurring. That the brain, during a speeded task like this, the brain may happily throw blood at a large portion of the brain, so that so that you have a lot of resources available to do the task. But the task may not use all of that blood flow. So the brain is rather, we think that the brain is rather profligate in its blood flow allocation. So we can use that as perhaps a way to look at different kinds of way to look at metabolic response. So perhaps this is a promising scheme for clinical applications. So these stable activation patterns are potential diagnostic of cognitive function. The stable timing is a potential diagnostic for neurovascular integrity. And this simple low-level task can be performed by many patients with disorders such as a traumatic brain injury or early Alzheimer's disease, although our experience is that we can't get we've tried a number of AD patients, and the ones who are too far gone can't do the task because they can't remember the instructions. So there are some limitations here. And the stable timing differences, as I mentioned, could reflect different metabolic demands. And a number of pathologies are suspected to be a marker for these pathologies is energy crisis in traumatic brain injury and in forms of dementia. Not enough oxygen is believed to be a marker for regions of the brain that are damaged. So if we're going to use this for as a biomarker. We want to make sure that it's stable for a long period of time. So we looked at stability in across periods of from a few hours to a few days to a few months using this structure. We scanned a subject first, and then we scanned them on one day twice, and then 3 months later we scanned them a fourth time. And we looked at the temporal differences, and we were rather surprised at how stable these HRF responses were. So here are some examples for both positive HRFs and some negative HRFs. And you can see that, so that we've overlaid the 4 different time points. And you can see that there just isn't a whole lot of difference between them. And as what we also discovered is that there isn't a whole lot of difference between the 3-month time point and the 3-hour time point. There's no significant difference in the errors. So the RMSE across the 4 session time points is generally much less than 0.1%. And the spatial average RMSE for all subjects is similarly very, very low. And it was also interesting to see that that the errors were not stationary, that there are greater errors during the hyperoxic peak or the anti-hyperoxic peak, the hypoxic peak for the negative HRFs. This has some interesting implications for the kind of noise mechanisms that are present in, so that there's a vascular component to the noise mechanisms as well as as probably an MRI component. So we also wanted to, recently we started wanting to study age-related effects. So we added a group of 14 elderly subjects aged 65 to 78, not quite, with a slightly larger number of females, and all subjects cognitively normal. And we folded this data into 2 sessions from the previous study, the ones where we scanned the 2 sessions that we got on the same day, yielding an age range between 20 and 78 and a total of 54 sessions. And we saw that rather immediately, that if we just looked at the mean values of these parameters averaging over the entire brain of each subject, we saw that there are some interesting trends. Clearest is this high frequency power fraction, which the mean high frequency power fraction seems to increase rather strongly with age, both for positive HRFs and for negative HRFs. There are also some weak trends for the peak amplitudes. The peak amplitudes show a slight tendency to drop with age for positive HRFs, and for negative HRFs, however, there's actually an increase in peak amplitude, in negative HRF peak amplitude with age. We don't entirely understand this trend yet. There's an increase also in the fraction of cortex that corresponds to negative HRFs. So it looks like older people have a more solid use of negative HRFs. There's also an interesting weak trend, barely significant, P of 0.055 for full width half max to decrease for the negative HRFs. So another thing we looked at was the spatial standard deviation. So essentially, if you look at the distribution of parameter across the whole of cortex and just evaluate the standard deviation, then we found that peak amplitude shows a very strong decrease, so that there's less spatial variability across cortex for positive for the positive HRF. And full width half max shows a very strong trend, a very very strong correlation with with age for the standard deviation of the full width half max. In other words, there's more spatial variability of the full width half max. And likewise, there's also more spatial variability of the high frequency power fraction. And these effects are also occur, the full width half max and high frequency power fraction also occur for negative HRFs, the spatial variability. Time to peak shows, interestingly, also an increase in spatial variability for negative HRFs only. So the most interesting of these effects are the full width half max and the high frequency fraction. So full width half max, it turns out, if you look at these distributions, is that young people show a single mode. Here's an example subject, one 29-year-old. Middle-aged person, these have broadened substantially, and maybe there's a trace of a second mode. And by the time you get out to older ages, then you start to see this bimodal distribution. And now if you average all of the people in our young group, less than age 40, our older group, greater than age 60, and the middle age in the 40 to 59 range, then you can see that if you average the normalized distributions together, that the distributions grow this second fast mode. So that's really interesting. And here are some examples of that show the character. So here's from our young subject, and you can see all the HRFs basically look very, very similar. But here are examples that show both the standard in this middle-aged example, there's standard HRFs, but then in the dashed lines, you see narrow full width half max. And these have, they don't have shorter onset time, but they do have narrower full width half max. And then in the older subjects, the effect becomes more extreme, and there also is perhaps a change in the onset time as well. So something is fairly dramatic seems to be happening with age. But do remember that only some of the older subjects showed this pattern towards narrow full width half max. Some of the subjects show their brains really look, even when they're older, they look like younger brains. Likewise, the high frequency fraction shows an increase with age in both in terms of area, the number of voxels, and the spatial variability of that high frequency fraction. So young people, their high frequency fraction is generally low all over their brains. By the time we get to middle age, they're getting splotches of this high frequency fraction. And then by the time we get to our older subjects, there's a lot of variability and a lot of high frequency fraction. So the power spectra suggest that this high frequency fraction corresponds to coherent modes. So here are all of the power spectra normalized to the low frequency component. Most functional MRI has a peak down below 1/10 of a hertz. That's sort of the standard HRF signature. but we have a number of our older subjects, 50 on up, that show these coherent peaks at high frequencies. And if you average in our groups of young, middle, and old, you can see how this power tends to pile up. And with age, the frequency seems to be pulling downward. So what do these trends mean? Well, let's put it into context of what's happening with age. We know that arteries and arterioles develop thicker walls and narrower lumens. It happens, all of these changes happen to all of us. There's also a certain amount of arterial rarefaction. There's a loss of arterioles and capillaries. It's rather widespread. And both of these effects are increased by hypertension quite substantially. And the consequence of these changes is that there's reduced arterial blood volume and reduced baseline blood flow. And what this might be doing, an interpretation that I favor, is that you're decreasing the inertance of these systems, the vascular system. Here is a much more complete linear model of the Peel mesh, which actually has a roughly hexagonal shape. So if you visualize that the entire cortical surface is tiled by these hexagons, then and all of their characteristics are fairly similar, quite stereotypic, so that you have many poles aligned with many zeros. And if you're decreasing the inertance globally, that might cause an increase in the bandpass frequency of these networks. And that might explain the decreased full width half max that's occurring. And it doesn't seem to happen everywhere, but it seems to happen in various patches of cortex. And I wonder if disruption of this Peel mesh could create certain high frequency parasitics, because you're unbalancing this network, this mesh of essentially linear elements, and create some high frequency components. So that completes my too long talk, and I started with an introduction to functional MRI, which has the basic notion that the brain lights up, functional MRI lights up the brain caused by a blood flow response. I showed you a little bit about how we model the hemodynamic response using this combination of simple flow and metabolism models coupled with a 1.5D model. And then I showed you a few examples of HRF measurements where we can essentially light up the whole brain, and now we're starting to use this approach to study age-related changes in the brain. I want to acknowledge the people who contributed to this. Jungwon Kim and Amanda Taylor were very strongly involved in this work. More recently, Nushin Fesharaki has been piloting the age-related work, and Keishan Mosby helps to coordinate our research, coordinate our scanning, and Valerie Pavlik in BCM neurology has been helping us with our trying to scan patients with age-related pathology. And I acknowledge my grant funding from NIH, both NINDS, National Institute of Neurological Disease and Stroke, and the National Heart, Lung, and Blood Institute. Thanks very much.
Thank you. That was really interesting, and I could see your hypothesis at the end makes sense. So we're ready for questions, And I'm wondering if anybody wants to either put questions in the chat box, or do you want to raise your hand or indicate that you would like to talk over the microphone instead? Okay, Michi Garrison.
Yeah, hi. Very interesting talk. Thank you very much. I was a little confused about the negative signal. I understood about brains, the electrical activity causes increased oxygen uptake, but I wasn't quite sure what kind of activity would cause a negative oxygen uptake. Can you explain that further?
Well, that's a good question, one that I don't have a really good answer to. We don't really understand. We understand the positive HRF quite moderately well, quite moderately well. We understand the negative HRF rather poorly. We know that there's the signaling mechanisms involved in the positive HRF are complex. They involve one of the primary movers is NO, and NO is a signaling mechanism, a signal produced by the astrocytes, and the astrocytes can sense tend to sense signaling because they are involved in glutamate recycling, and so there's a fairly detailed understanding of that process. We know that neuropeptide Y is involved in the vasoconstriction process, and we've been able to, the work of Anna DeVore in particular has demonstrated the sequence that goes on in the negative HRF, but we really don't understand which synaptic mechanisms, and there's no equivalent to proximal integration for the vasoconstriction response.
But you were doing different tests to elicit responses. So were there some tests that elicited a rapid positive response, and then other tests resulted in a negative response?
That's the whole beauty of the approach, is that it's just one task that evokes both the positive and the negative responses that cover about 88%, 87% of cortex. The variability in the response corresponds to, no doubt, to the variability of how the brain does its processing of that single task, which I suspect is actually a spatio-temporal process, because it's actually very, if you think about it, generating a blood flow response is not that easy, because what do you think about it? Getting blood to flow to various portions of the brain isn't easy, because the brain is basically wrapped in Kevlar. It's a fluid. It's immersed in water, CSF, that's wrapped in Kevlar, which is the durameter, and then pressurized slightly and encased in a bowling ball skull. And so how do you drive blood into a pressurized container? It's actually not straightforward at all. And we've been arguing about how this trick happens for quite a while. And it may be that I often wonder if the negative bold response is actually simply a way of robbing Peter to allow Paul to be paid. In other words, the hemodynamics demand that something has to move out of the way. in order to get more blood in place. And so somebody's got to give. This whole spatial process, which may involve wave formation. There's been some work about the generation of traveling waves in the cortical in cortex, probably in the CSF, and there's been a lot of work about how these blood flow processes are involved with the motion of CSF, which is very intimately involved with sleep and cleansing of metabolites from the brain. And they have to be related to the hemodynamics. The hemodynamics has been shown to drive the CSF motions. So it's a pretty intricate process getting blood into the brain.
Oh, great. Thank you very much.
Any other questions? All right. Well, thank you very much again, David, for your presentation, and also thank you for your contribution to our further understanding of the body and perhaps of aging mechanisms. So you're doing good work.
Thank you.
And I'll say good night then to everybody. And as I mentioned before, we're going to have a joint virtual meeting with the MIT alumni group on April 26th. I will send out a message to my entire mailing list, and hope to see some of you on that. I will not be the host. It'll be hosted by MIT. And also there's a small cost involved. The MIT Alumni Association charges, and IEEE members will get a discount that amounts to ultimately paying, I think, $5.28.
Not too much.
All right. Well, thank you again, and I'll stop the recording.